So now that we have an idea of how genes are expressed, we need to ask the question of how is it decided when a gene should be expressed, so that an organism doesn't waste a whole bunch of energy producing gene products that aren't even needed. This is where we begin the subject of genetic regulation. In prokaryotes and eukaryotes alike, we have antisense RNAs, microRNAs, and riboswitches that are involved in regulation of gene expression. But prokaryotes have another strategy too. They organize collections of related genes into operons. And these operons contain control mechanisms that ensure that genes are active only when their products are required. This coordinated set of genes are regulated as a single unit, and they're either inducible operons or repressible operons. An inducible operon is an operon that's turned on by the substrate of the enzyme for which the structural genes code. That is, they're only turned on when the substance that needs to be broken down is present, so those enzymes are only manufactured when they're needed. Repressible operons contain genes that code for anabolic enzymes. In this system, several genes in a series are turned off by the product that's synthesized by the enzyme. So that is, they're on all the time, and if there's too much product present, then they'll shut down. Let's take a look at a couple of examples. We'll begin by looking at the lactose operon. This is a great model for inducible gene regulation. The system is found in Escherichia coli, and it's involved in regulating lactose metabolism. First of all, it's important to understand that glucose is the preferred energy source for these bacteria. However, if glucose is not present, then the bacteria can survive on lactose. So if glucose is present, this whole system is shut off. It's only turned on when glucose is absent and lactose is present, so that the bacteria can switch to using lactose as an energy source. The operon has three important features, whether it's inducible or repressible. The regulator is a gene that codes for a protein that's capable of repressing the operon, or coding for a repressor protein. So in this image, here is our repressor protein. Somewhere upstream of this operon is a gene that codes for this protein. That is the regulatory gene, and it's not shown here in this picture. Then there's going to be a control locus on the operon, as well as a structural locus. The control locus is involved in controlling the expression of the gene, whereas the structural locus contains the three genes that are expressed when this operon is activated. So within the control locus, we have both a promoter and an operator. The operator is sort of like the operator on a train track. When you go up to the operator and say, hey, can I get onto the tracks or not? He's going to say yes or no. Now, as you'll recall, RNA polymerase is the enzyme that's involved in transcribing prokaryotic genes. It's going to try and get onto the gene to transcribe it. It starts by coming to the operator region. If the operator region is blocked by this repressor protein, then RNA polymerase cannot move down the tracks. So keep in mind, this is an inducible operon, so it's usually in the off mode. It doesn't initiate transcription unless lactose is present and glucose is absent. So if lactose is present, it binds onto the regulatory protein and it removes the regulatory protein from the operator region, so RNA polymerase can jump onto the operator and transcribe the gene. The operon contains three genes, structural genes, that code for proteins. Each of these three proteins are involved in the process of digesting lactose. One of them breaks lactose apart into its monomers. Another of them allows it to be transported across the membrane, so on and so forth. Once lactose has been digested, however, there's less lactose present. Without the lactose present, then the repressor protein is free to bind again to the operator region. And there, it once again is going to block transcription of the three genes necessary to digest lactose. So again, this system is generally in the off position. When lactose is present, it removes the repressor, allowing transcription of the 3 structural genes. When there's no longer lactose present, the repressor protein is released, it can clip back onto the operator region, and there'll be no transcription of those 3 gene products. Naturally, this can be tough to follow. Again, I think here, a moving picture will do us a lot of justice, so let's check out an animation.
The expression of genes can be transcriptionally regulated. Such regulation is a critical feature of both eukaryotic and prokaryotic organisms. One of the classical systems used to investigate transcriptional regulation was originally investigated by Jacob, Monod, and Pardee. Their studies involved the transcriptional regulation of the genes used to ferment lactose in the bacterium Escherichia coli. 3 genes clustered together on the chromosome are required for the bacterium to utilize lactose. These were determined to be the LAC-Z gene that codes for the enzyme beta-galactosidase, the LAC-Y gene that codes for a lactose permease, and the LAC-A gene that codes for the enzyme galactoside transacetylase. The LAC-P region is the promoter that is needed to transcribe the LAC-Z, LAC-Y, and LAC-A genes as a single polycistronic mRNA. The LAC-O region is an operator site that is involved in the transcriptional regulation of the LAC operon. The unit consisting of the LAC promoter, LAC operator, and the LAC-Z, LAC-Y, and LAC-A genes is called the LAC operon. Located near but not in tandem with the LAC region is the LAC-I regulatory gene that codes for an mRNA that is translated to produce a protein referred to as the LAC repressor. In the absence of lactose inside the cell, the LAC repressor protein is active. In its active state, the LAC repressor recognizes and binds to the LAC operator site. When the LAC repressor is bound to the LAC operator site, RNA polymerase is prohibited from recognizing the LAC promoter, and the LAC-Z, LAC-Y, and LAC-A genes are not transcribed. When lactose enters the cell, a small amount of it is converted to allolactose via beta-galactosidase. Allolactose binds to the LAC repressor. This causes a conformational change in the LAC repressor protein that prevents it from binding to the LAC operator site. Inactive LAC repressor is unable to bind to the LAC operator site. Without an active LAC repressor bound to the LAC operator site, RNA polymerase is able to bind to the promoter, and a polycistronic messenger RNA is transcribed that includes the LAC-Z, LAC-Y, and LAC-A genes. This polycistronic messenger RNA is then translated to produce the beta-galactosidase, lactose permease, and galactoside transacetylase proteins. Therefore, the induction of the LAC operon enables the bacterium to efficiently transport lactose into the cell and metabolize it.
A repressible operon is essentially opposite in function. It has all the same components, the promoter, the operator, and the structural genes. Normally, the operon, however, is in the on mode, and it'll be turned off when a nutrient is no longer required. The system we often use as an example for this is amino acid synthesis. So we'll look at the arginine operon and the tryptophan operon. In this case, the operon is usually on, which means that there's no repressor bound to the operator sequence. Means that RNA polymerase can jump onto that sequence and transcribe the structural genes. In this case, the structural genes end up in the production of these certain amino acids. When these amino acids are being produced, they're consumed, and so there is no excess. However, should there become an excess of the amino acid, then it will bind to the repressor protein. In this case, binding to the repressor protein activates the repressor protein. So it's called a co-repressor because it's activating the repressor protein so that that repressor protein can now bind to the operator region and prevent the RNA polymerase from jumping on the promoter and transcribing the genes. Once there becomes not enough of the arginine or tryptophan, it falls off of the repressor is moved from the operator region, and once again RNA polymerase can transcribe the genes as needed. Let's take a look at a quick animation of the trip operon.
In the bacterium Escherichia coli, a group of 5 genes code for enzymes required to synthesize the amino acid tryptophan. All 5 genes are transcribed together as a unit called an operon. An operon is a group of genes that is under the control of a single operator site. A regulatory protein called a repressor can bind to the operator site and prevent transcription. When tryptophan is lacking in the environment, the repressor is made, but it is unable to bind to the DNA and block transcription. RNA polymerase binds to the promoter site and then proceeds down the DNA, transcribing the genes for the tryptophan biosynthesis enzymes. When tryptophan is present in the environment, the organism no longer needs to make tryptophan. Tryptophan binds to the repressor and activates it. The activated repressor now binds to the operator located within the tryptophan promoter and blocks transcription.
The naturally occurring nutrients are not the only agents capable of modifying gene expression. In fact, many of our antibiotics are targeted at affecting gene expression by interacting either with the ribosomes or transcription or the translation process. Certain drugs are going to react with the DNA, RNA, or ribosomes and alter their genetic expression. Growth of the infected agent will be inhibited by blocking its protein synthesizing machinery selectively. If it doesn't have protein synthesizing machinery, then it can't express any of its own genes, and thus it can't live. So drugs that inhibit protein synthesis exert their influence primarily on transcription and translation. Here are some of the antibiotic strategies. Antibiotics often target the ribosome. Remember that the prokaryotic ribosome is different than a eukaryotic ribosome. So with an antibiotic, we can inhibit the ribosomal function of the prokaryote without actually affecting our own ribosomal function. Without ribosomes, they cannot synthesize proteins. Now, there's a little bit of a problem with the antibiotics that target ribosomes because in humans, you'll recall mitochondria, they have ribosomes that are of prokaryotic nature. So sometimes we'll see issues with mitochondria in that antibiotic treatment. Now, rifamycins used for tuberculosis bind to RNA polymerase. If RNA polymerase is bound, it cannot bind to the gene and transcribe that gene. And you'll recall that RNA polymerase in bacteria are slightly different than those in eukaryotes. So the rifamycins are more active against the bacterial RNA polymerase than they are ours. Erythromycin is another antibiotic. It works by preventing prokaryotic translation. It does this by interfering with the attachment of messenger RNA to the ribosomes. If the messenger RNA can't attach to the ribosomes, then no translation can happen. Tetracycline has a different strategy. It binds to the ribosome in such a way that it prevents elongation of the polypeptide chain. So as you can see, there are a plethora of different ways in which we can interfere with the processes of transcription and translation in order to control microbial growth. Let's have a quick concept check. What is the term for the DNA sequence that is bound by regulatory proteins in bacteria? So which sequence on the gene is where the regulatory protein binds? Well, the operon is the whole system. The promoter is where the RNA polymerase would originally bind. Exons and introns, they just don't exist in bacteria, so that leaves us with the operator. The operator is the region of DNA where regulatory protein binds in either inducible or repressible operons. So now we've explored some of the many ways that gene expression is controlled. Fascinating stuff. Next, we'll move on to look at mutations, changes in the genetic code.